It is my pleasure to introduce Adriana Romero-Olivares, joining us as a brand new assistant professor from the Department of Biology at New Mexico State University. Adriana did her PhD at the University of California at Irvine with Kathleen Treseder, and then recently completed a postdoc at the University of New Hampshire with Sarita Fry. And Adriana has been incredibly productive publishing, including multiple papers in top journals from her master's thesis, her dissertation, and also her postdoc work. She also received several prestigious grants and fellowships during her dissertation and postdoc, including a UC Mexis fellowship that funded her entire PhD and a Diversity and Innovation Scholar Fellowship at UNH. And Adriana is also very active in the broader scientific community around diversity, equity, and inclusion, and regularly gives talks, participates in outreach, and contributes to written content on these issues. So with that, let's give her a warm virtual welcome as she tells us about the exciting work she's doing on the most charismatic organisms on the planet, fungi.
Thank you so much for the invitation. I'm really excited to be speaking here today. As Krista said, I am a soil microbiologist, but I do have a very strong bias for indeed the most charismatic organisms, fungi. And I'm really interested in how soil microbes are responding and adapting to climate change and how that consequently affects the cycling of carbon and the emission of greenhouse gases into our atmosphere. I've been exploring these topics since I was a grad student and now in my lab at New Mexico State University, which is in the land of the Apache. And I do want to point out that my pronouns are she, her. So today I'm going to be talking about microbial responses to climate change from tiny genes to big ecosystems. So when I decided to study biology, I did it because I was fascinated by landscapes like this one with a lot of plants and presumably with a lot of animals. And back then, I never really thought about what was beneath all of that visible biodiversity. But as I moved forward in my degree, I started to learn about the importance of soils and its very large invisible biodiversity and how essential this biodiversity is to keep ecosystems working. So for example, in this image, you can see that in a relatively small piece of land, there's approximately 50 kilometers of mycelium of around 500 different fungal species. There's 100 billion bacterial cells of approximately 10,000 different species. There's 500 meters of roots of approximately 10 different plant species. And you see where I'm going. There's a lot of biodiversity in the soils that in most cases we cannot see. And in my research, I focus in most cases in fungi. More specifically in saprotrophic fungi. So soil fungi are fungi that live all of their life or a part of their life in the soil. And there's a lot of soil fungi. So in a single gram of soil, there can be thousands of different species of fungi. And if you're lucky, sometimes you can see them. So for example, here you can see a piece of wood that it's colonized by a very thick mycelium. So all of these white threads right here are fungal mycelium. And then here you can see a picture of the traditional mushroom, the one that everyone thinks about when someone says the word fungi. And on this other one, this other picture, we can see a different type of fungal fruiting body. It's not the traditional quote unquote mushroom, but it's still a fruiting body. It's still a fungus and it's still visible to the naked eye. But the great majority of fungi are microscopic their entire life cycle. Sometimes we can see them if we isolate them with microbiological techniques, like all of these cultures that you can see in this picture. But some other fungi we have never seen, but we know that they exist thanks to molecular techniques, which I use a lot in my research. And to jump into my research topic a little bit more, as you can imagine, soil fungi, even though they're invisible, they are extremely vulnerable to changes in the environment. but because they're invisible, they're very difficult to study. So for example, in this picture, you can see that the right side of the picture looks impacted, and the left side of the picture looks quote unquote healthy. You can say that because of what you see on the ground. You can see that the right side of the picture has eroded land versus the left side has grass. You can see that one side of the tree doesn't have any leaves versus the left side, which does. So based on those things that you can see, you can say that probably the left side ecosystem is impacted, whereas the left side is not. You can assume things about the microbial community based on that, but you can't really say anything else. You can't really see anything, so you can't really say much. So with this, I'm trying to say that there's certain things that us microbiologists simply cannot do with our study system. Some other things that we cannot do is, for example, we cannot follow fungal life cycles because it is impossible to count the progeny of soil fungi, especially the microscopic soil fungi. There are no historical records on the distribution of fungi except for a very few mushroom species, and these records are specific to certain parts of the world. And then there's many other reasons why studying microscopic fungi is really difficult in ecology and evolution. But despite these difficulties, it is very important that we study fungi because fungi are mediators of biogeochemical cycles. They're responsible for keeping nutrients and elements flowing in our ecosystem, specifically between land and atmosphere. So for example, in this very simplified picture of the carbon cycle, you can see that with sunlight and carbon dioxide, this plant photosynthesizes and produces a lot of biomass in the form of leaves and in the form of wood. Eventually, that biomass falls off and becomes plant waste onto the ground. When it's in the ground, decomposer fungi are ready to start decomposing. So through the process of decomposition, fungi acquire some of that biomass, some of those nutrients to grow and to produce their own plant biomass. Another portion of the carbon is organically bound in the soil. And then another portion of that carbon, and as a byproduct of the fungus metabolism, is released as carbon dioxide into the atmosphere. And you can see here how that completes one cycle of the carbon cycle. So again, very simplified carbon cycle picture, but I wanted to make sure that we are all on the same page on what I mean when I say that fungi are mediators of biogeochemical cycles. And I do want to make a parenthesis and say that there's another super important group of fungi, the mycorrhizal fungi, which in most of my work I don't look into, but they are regardless super interesting and have a very important role in our ecosystems as well. So all of these is very important because soils are not only the home of soil fungi and of other microorganisms and organisms, but they're also a carbon storehouse. So there's around 1,500 billion tons of carbon stored in the soil on top of the 750 tons of carbon already available in the atmosphere. So there's twice as much carbon stored in the soil compared to what's already available in the atmosphere. So because soil is a storehouse of carbon, it can also be a source of carbon dioxide. And it is the best that carbon stored in the soil stays there, because if it's released into the atmosphere, then carbon dioxide concentrations would increase. And as we all know, carbon dioxide is a greenhouse gas. So in a few words, if a lot of the carbon that's stored in the soil is released into the atmosphere via decomposition, then the concentration of carbon dioxide in the atmosphere would increase, the greenhouse gas effect would worsen, and that would feed back global climate change. And all of this is really important because the molecular machinery that microbes use to decompose and store and return carbon dioxide in the atmosphere is extremely sensitive to environmental change, any sort of environmental change. So impacts to soil fungi or impacts to the environment, more generally speaking, have an impact in microbial metabolism. And this can result in a lot of different things. For example, it can result in millions of dollars lost in crops, as I just mentioned, increases in greenhouse gas emission, and it can even result in changes in the microbial community composition and abundance that could result in the emergence of pathogens of plants and animals. So with all of this in mind, as a graduate student, I went to the boreal forest in Alaska in the land of the Tanana. I was especially interested in studying these ecosystems because boreal forests store approximately 510 billion tons of carbon in the soil. And changes in temperature brought by global climate change have been really dramatic in these ecosystems at these latitudes. So in this map, this is a map of the state of Alaska. I worked in a place called Delta Junction. You can see it here with a star. And according to this map, all of the state of Alaska, including the area of Delta Junction, is significantly affected or has been significantly affected by changes in temperature. So specifically Delta Junction, you can see here, it has experienced an increase of 1.7 to 2.2 degrees Celsius in the average annual temperature compared to the past 50 years. So I conducted research in this field experiment that had been ongoing for over 10 years. And it's a really simple field experiment. It consists of 5 greenhouses in the middle of the forest, just like this one in the picture. And you can see where each one of the different greenhouses is in this other picture from above. And these greenhouses passively warm the soil and the air inside them. They increase the inside air temperature by 1.5 degrees Celsius. So basically around the increase in temperature that Delta Junction is experiencing. And it increases the temperature of the top 10 centimeters of soil by 0.5 degrees Celsius. Evapotranspiration is higher inside the greenhouses, and this causes the inside of the greenhouses are 22% drier compared to the outside. And something that's important for me to mention is that the tops of the greenhouses are removable. So we remove the top of the greenhouses from September to May, and then put them back in place from May to September. And maybe you're thinking, wow, 0.5 degrees Celsius is really nothing. Like, how can that have an impact inside the greenhouses? It's really not a big difference. So here, you can see how different the inside of the greenhouses look compared to the outside. So here you can see that there's a lot of grass, a lot of mosses. There's a few mushrooms right here, a lot of plants. And then inside of the greenhouses, you can see that it looks extremely dry. There's still one mushroom here though, but you can see that there's no mosses at all. There's very few plants. So even though the increase in the top 5 centimeters of soil is only of 0.5 degrees Celsius, you can see how drastic that increase in temperature, how drastic the difference is between the control and the treatments. So there were a few questions that I wanted to address in this experiment. I wanted to know what's happening with the soil fungal community under warming and drying. Where are they allocating resources, and how does this reflect in the cycling of carbon? So to answer these questions, I carried out 2 different experiments. The first one was soil metatranscriptomics, and the second one was measuring decomposition rates. So for soil metatranscriptomes, I isolated RNA from soils, from the control and from the treatment. And then I analyzed 2 different sets of transcripts. Transcripts that were transcribed from genes involved in metabolic maintenance, and transcripts that were transcribed from genes involved in decomposition genes. So I'm going to refer to the metabolic maintenance genes as COG genes and the decomposition genes as Casii genes. And then the decomposition experiment was really simple experiment. I really, really love litter bag experiments, and that's one of the experiments that I conducted. So for those of you not familiar with litter bag experiments, they're really simple. So you can see a litter bag right here, and It's really simple. Again, you just collect leaves from the trees. In this case, I collected spruce needles because spruce are the plant that it's more abundant around my field site. So I collected 2 grams of spruce needles, and then you put them inside a litter bag, and then you deploy the litter bag on the forest floor and collect them throughout time. And then you measure decomposition based on weight loss. It's a very simple but very powerful experiment. So what did I find? Okay, so I found that warming and drying favors melanized, pathogenic, and desiccation tolerant fungi, such as crust fungi and lichens. So Silaria, for example, was by saying that these fungi were favored by warming and drying, I mean that these fungi were the more active under warming. And by more active, I mean the ones that were transcribing the most. So again, a lot of melanized fungi like this one, Silaria, Janula, which is also a lichen, crust fungi like this one, and then a bunch of pathogenic fungi that can also be saprotrophic, like penicillium, for example, and a bunch of fungi from the order of the Diaprotalis. So I also found that metatranscriptomes revealed a trade-off in resource allocation. And before showing you results, I want to walk you through the graphs. So on the x-axis here, you'll see metabolic genes, COG genes, and then decay genes, which are also known as Casii genes. And then in the y-axis, you'll see total gene transcripts. This graph is for COG genes, and this is for decay genes. And then the control bars are in gray, and the warm bars are in black. And as you can see, the trends are the exact opposite, meaning that under warming, microbes allocate more resources to cell metabolic maintenance and less resources to decay. Whereas in control conditions, you see the exact opposite. And I do want to point out that transcripts are just that, they're transcripts. We can be sure that all of these transcripts, which are messenger RNAs, are going to mature and be translated into enzymes. And we also can't be sure that these changes will have an impact at the ecosystem scale, but it was still super neat to find this trade-off in resource allocation. However, our field decomposition experiments supported our metatranscriptomes findings. And I'm gonna show you what I mean by that. In this graph, in the x-axis, you'll see decomposition times. So basically the times where I collected the litter bags from the forest floor. And then in the y-axis, you'll see mass remaining as percentage. And in this case, the bars are, the white bars are control and the black bars are warming. So what you can see is that for all collection times, the percentage mass remaining was always higher in warm plots. So this means that decomposition in warm plots was lower compared to decomposition in treatment plots. So indeed, what we found in our metatranscriptomes data, that under warming, microbes are investing less in decomposition, was supported by our field decomposition experiment, because there's less decomposition under warming compared to control. So to answer the specific questions that I wanted to answer, what's happening with the soil fungal community under warming and drying? Pathogenic, melanized, and desiccation-tolerant fungi were more active under warming. Where are they allocating resources? They're allocating more resources to cell metabolic maintenance and less resources to decay. How does this reflect in the cycling of carbon? There's less decomposition under warming. So are these results good or bad? And the answer is that I guess it's both. So first, it looks like it's good because there's potentially less CO2 that's being returned into the atmosphere. If you assume that less decomposition means less CO2, then that's what we think it's happening. But shifts in the fungal community are really concerning, especially the increases in pathogenic fungi. And we have to keep in mind that other microorganisms and plants acquire their nutrients from fungal decomposition. So less decomposition also means less nutrients available for plants and animals. I also want to point out that our results are not unique to our field experiment, and I conducted a meta-analysis over here published in Cell Biology and Biochemistry, where I saw that many other warming experiments around the world are reporting similar results, specifically in the sense that it looks like warming experiments reduce, or we can see a decrease in CO2 as the duration of warming increases. But I also want to point out that we found a lot of nuances in our results, and you can read a little bit more about those in my 2017 paper. So even though there was overall less decomposition, there was an increase in the decomposition of a very specific carbon fraction. So it looks like it's too soon to tell if our results are good or bad, but what we know is that global climate change is likely to affect the fungal community with consequences at the ecosystem scale, such as disruptions to the carbon cycle. And along these same lines of research, I continued with my postdoc work, but this time I explored fungal ecological traits a little bit more, their role in decomposition under global climate change and consequences to the carbon cycle. I really love the carbon cycle. So it is well known that soil carbon and nitrogen dynamics in the soil are tightly linked. And what links these two together is the fungal community, and impacts to either one. For example, changes in the concentration of soil nitrogen or changes in the chemistry of organic carbon can directly affect decomposition and consequently CO2 emissions. And the chronic nitrogen amendment study at Harbor Forest, which is in the land of Nipmuc, is a perfect example of this relationship. So this experiment has been ongoing for 32 years. It's actually one of the longest LTRs around the world, and it consists in chronic fertilization of nitrogen in the form of ammonium nitrate. So this experiment is somewhere around here. And this experiment started in the 1970s and the 1980s because nitrogen pollution was a huge problem in the northeast of the US. So in all of this area around the 70s and the 80s, they were experiencing a lot of issues related to pollution, nitrogen pollution. So now nitrogen pollution is not a huge problem in that area. Thanks to a lot of environmental policies that were established in the 1990s, which helped decrease nitrogen pollution. But this experiment continued because nitrogen pollution is a problem now in other places in the world, included some areas in the US, for example, in the Southwest right here, where I live right now. And one of the main observations in this experiment is that as the concentration of nitrogen in the soil increases, the amount of organic matter increased as well, meaning that the higher the nitrogen concentration, the lower the decomposition rates. In this graph, in the x-axis, you'll see the nitrogen addition in kilograms per hectare per year, and this is the control, and then these are the two treatments. And then in the y-axis, you'll see soil carbon stocks as kilograms per square meter. And then you can see the organic horizon in light brown and the mineral horizon in dark brown. And what you can see is that carbon stocks are higher in treatments compared to control. So these two are basically higher compared to this one. And this is especially noticeable in the organic horizon, which are the first 10 to 15 centimeters of forest floor. And this is the horizon that I'm most interested in studying because most fungi inhabit that horizon, most soil fungi. So I was wondering, what may be the mechanisms behind this accumulation of organic matter? Why, when nitrogen in the soil is high, carbon stocks are also high? And one of the good things about working in these very long field experiments is that a lot of people have done a lot of work before you. So you have a lot of hints that you can use in your research to inform the directions that you're going to take. So I already had a few hints of what may be going on and what may not be going on. So I already knew that it didn't have to do with the amount of leaves that were falling from the trees, because the same amount of leaves were falling from trees in the control plots and in the nitrogen plots. I also knew that it didn't have to do with the chemistry of the leaves, because when the leaves fell to the ground, their chemistry was exactly the same. So there were no changes in the chemistry of the leaves when they fell onto the ground. I also knew that it probably had to do with fungal community composition, because a previous study had already identified that the fungal community shifted in the nitrogen plots compared to the control plots. So the specific questions that I wanted to address here were, how do gene frequencies involved in the processing of carbon and nitrogen can help us understand what's going on at the chronic N field study? And could we use fungal trait-based groups paired with gene frequencies to predict responses of the fungal community to global climate change? So to answer these questions, I used Microcosm, which is a wonderful database. This database has approximately 1,000 fungal genomes. And then I grouped all of these genomes or all of this taxa using another online tool known as FunGuild. So this tool basically categorizes fungi by trait-based groups. Then I calculated gene frequencies of gene families involved in the uptake of nitrogen and the decomposition of carbon. And by uptake of nitrogen, I mean genes that codify for molecules involved in processing of nitrogen, such as ammonium transporters, nitrate transporters, and amino acid permeases. And by decomposition, I mean genes that codified for molecules that are involved in the processing of carbon, such as ligninases, cellobiohydrolases, beta-glucosidases, lactic polysaccharide monooxygenases. And I do want to point out that I focused on gene families, not individual genes. I want to make a parenthesis before I show you some of our findings, because I want to make sure that, I go over the different trait-based groups in FunGuild. So there's 4 major trait-based groups: trophic mode, which categorizes fungi as symbiotrophs, saprotrophs, or pathotrophs. The next category is functional guild, which categorizes fungi based on the resource they exploit: animal pathogens, ectomycorrhizal fungi, endophytes, plant pathogens, undefined saprotrophs, and wood saprotrophs. And then the other trait-based group is morphology, which categorizes fungi based on their phenotype or their morphology. And this is my favorite category. You can see all of the different categories here. So agaricoids, boletoids, corticioids, gasteroids, microfungi, polyporoids, and yeast. And then the 4th category is a rot type, which categorizes fungi based on their ability to decompose certain types of carbon: brown rot, white rot, or soft rot. For example, this organism right here, its scientific name is Pleurotus ostreatus, is also known as oyster mushroom. Its taxonomy, it belongs to the Basidiomycete in the order of the Agaricales. Its trophic mode is saprotroph, functional guild, wood saprotroph, morphology, agaricoid, rot type, white rot. And then based on its gene frequency, we know that it has a very low nitrogen uptake gene frequency, 1.08, and very high organic matter decomposition gene frequency, 12.17. So I basically did this for approximately 100 different fungal taxa. I'm sorry, 1,000 different fungal taxa. Okay, so these graphs, before showing you the results, I'm going to walk you through them too. So in the x-axis, you'll see nitrogen uptake gene frequency as number of genes per 10,000 genes. And then in the y-axis, you'll see organic matter decomposition gene frequency as number of genes per 10,000 genes. And then you'll see 4 graphs for each one of the different trait-based categories: trophic mode, functional guild, growth morphology, and rot type. So what we found was a difference in the distribution of fungal trait-based groups based on gene frequency of organic matter decomposition versus gene frequency of nitrogen uptake. We can see that we get different resolutions depending on the grouping that we use. So trophic mode offers little resolution right here, especially compared to the other three trade-based categories. But in general, we can see that ectomycorrhizal, right here, ectomycorrhizal, boletoids, and brown rots, they have low nitrogen uptake and low organic matter decomposition gene frequency. So they fall in this quarter right here on the corner. We also see that wood saprotrophs, polyporoids, corticioids, agaricoids, white rots have very high organic matter decomposition gene frequency, but low nitrogen uptake gene frequency. Then we see that yeast and animal pathogens have high nitrogen uptake gene frequency and low organic matter decomposition gene frequency because they fall in this quarter. And then we have the super fungi, the ones that have high nitrogen uptake and high organic matter decomposition gene frequency, which are the saprotrophs, the microfungi, and the soft rots. And of course, there are overlaps between groups because some fungi belong to more than one functional group, and we discuss all of these nuances in the paper. But once we had an understanding of how functional groups differed in their potential to uptake nitrogen and decompose organic matter, we reanalyzed the data in this paper. So this paper is this one right here, is the one that I mentioned a few slides ago, that it reported a shift in the fungal community in response to nitrogen. So this work was RNA-based, and it happened on the same field site. And the way that we reanalyzed the data was by calculating nitrogen response. It's a very simple formula that we came up with. So it's really simple. We basically counted the number of transcripts that we got for a specific taxa in the treatments, and then we divided the number of transcripts that we got for that same taxa, but in the control plots. And I'm gonna walk you through these graphs before showing you the results. So in the x-axis, you'll see nitrogen response. Positive values means an increase in abundance, meaning that there were more transcripts in the nitrogen treatments compared to the control. And then negative numbers means a negative nitrogen response, meaning that there were less transcripts in the treatment compared to the control. And then in the x-axis, you'll see in the y-axis, you'll see all of the different trade-based categories. And then bars are average nitrogen response plus minus standard error. And significant results are noted with asterisks. So what we found is trade-based categories that have positive nitrogen response have high nitrogen gene frequency and some very low organic matter decomposition gene frequency. So if you recall 2 slides ago, I showed you a few different functional groups that fell on the high nitrogen quarters. And what we can see is that those same functional categories are the ones that have a positive nitrogen response: animal pathogen, endophytes, general saprotrophs, and yeast. And if you're wondering, so what? Why is this important? Well, this linked gene trait framework, we see that there's potential for predicting how the fungal community will respond to environmental stressors based on their gene frequency and trait-based grouping. We need to figure out a few things and refine a few details. For example, we only got significant differences on the highest nitrogen treatment and not so much on the lowest one. But again, it looks like this type of work has a lot of potential for predicting fungal responses. And really quickly, I want to point out that gene frequencies by taxonomical categories do not have predictive power. And this potentially calls for a shift on the way that we analyze data in microbial ecology, because historically, most of the work has been taxonomically based, especially for the past couple of decades, because we figured out how to analyze a lot of samples using high throughput sequencing. But we are at a point where we can start sort of like shifting or linking taxonomic categories with gene frequency. So anyways, I'm going to show you. Okay, so if we do the exact same analyses, but using taxonomical orders instead, so nitrogen response on the x-axis and orders on the y-axis, we see that the hypocrealis and the ketotrialis have a positive nitrogen response. But then if we look at the gene frequencies of these groups, we see that the hypocrealis have like a mediocre nitrogen uptake gene frequency. The ketotrialis are somewhat on the higher end of nitrogen uptake, but they're not really significantly different to the dophitialis or the helotialis. So how do gene frequencies involved in the processing of carbon and nitrogen compare and contrast among different fungal trade-based groups? There's a differential distribution of fungal trade-based groups based on gene frequency of organic matter decomposition versus nitrogen uptake genes. Could we use fungal trade-based groups paired with gene frequencies to predict responses of the fungal community to global climate change? So it looks like we could, but we need more research linking trade-based groups with gene frequency and field observations to determine if it's a successful approach that we could use to predict responses. And as I mentioned, we also need to tease apart nuances. For example, why were our results only significant at N150 and not N50? And of course, it's important to remember that gene frequencies are a reflection of potential to uptake nitrogen and decompose organic matter. So we need more integrative research to validate these types of frameworks. In the last few minutes of my talk, I want to spend some time talking about one of my ongoing projects and share with you some exciting preliminary findings. So this was also a project of my postdoc when I was a diversity and innovation scholar. And I am now working in another field experiment also at Harbor Forest. It's called the Soil Warming and Nitrogen Deposition Field Experiment. We refer to it as SWAN for short. And this field experiment consists of plots that are actively warmed with heating cables, plots that are fertilized with ammonium nitrate, and plots that are both warmed and fertilized, and then plots that are left as is, which serve as controls. And you can see the setup of the field experiment here. So heated plots are in yellow, control plots are in blue, and so on. This field experiment has been ongoing for 10 years too. So it's part of the LTR at Harbor Forest. So in this field side, I was really interested in exploring volatile organic compounds and how their emission and profiles are affected by environmental stressors that we're expecting to see with global climate change, such as warming and nitrogen. So for those of you not familiar with volatiles, volatile organic compounds are organic chemicals. They vaporize at room temperature, and there's many different types of VOCs, some of which are ecologically important. And microbial VOCs have not been deeply explored in microbial ecosystem ecology, and they were recently listed as a grand challenge of terrestrial ecosystem ecology because of how little we know about them and how complex they are. So, a very straightforward example of a microbial volatile is the smell that we get when it starts raining. So that is a volatile that it's produced by a bacteria. So the smell of wet soil when it starts raining, that is a microbial volatile organic compound. So this project was a exploration type of study. And I wanted to know, do soil volatile organic compound profiles change under warming and nitrogen fertilization? And then I also wanted to explore the question, what is the role of volatile organic compounds in microbial communities under global climate change? So to answer these questions, I collected VOCs from the SWAN in 2018 and 2019 using this setup that you see here. I use these adsorption tubes collected to this machine, and then you can see the tubes right here. So these tubes adsorb the VOCs that are coming from the soil. And then using GC mass spec and specialized software, we determined which VOCs we found in control versus treatment, and we were also able to calculate emission rates. Okay, so these are brand new results. In these graphs, in the x-axis, you'll see treatment. This is control. This is heated or warming, however you want to call it. This is the nitrogen, and then this is the interaction. And then in the y-axis, you'll see total VOC emission as micrograms per square meter per hour. Then you'll see different plots per month, June, August, and October, and then per year, 2018 and 2019. And then you see all of the different VOC classes here in different colors. This is what we found. So we found that there was a lot of variation in the VOC emission depending on the year and depending on the month. We found that in August and October of 2018, the emissions in treatments were significantly higher than controls, but we didn't see that was a consistent pattern. So for example, in 2019, in August and October, we didn't really see any significant differences in the VOC emission rates of treatments compared to control. So to visualize these results, I conducted an NMDS. I wanted to see if maybe there was some sort of like clustering going on, even though our previous analyses had shown that there was probably no treatment effect. And indeed, we didn't really see any clustering based on treatment. So you'll see the different treatments in different colors, and then you'll see the different months in different shapes. And we see somewhat of a clustering based in year, but it's really not that strong. And indeed, once we see, once again, using this other clustering method, we see that VOCs are present in most of the controls and treatments alike. So these right here is for 2018, this is for 2019. And if you pay attention to this number in the middle, so these are the number of VOCs that are present in control and treatments. And you can see here that 91.6% of the VOCs are found in controls and treatments alike. And then you can see something similar in 2019. These are the total VOCs that we found in each year. This subset of VOCs were specific for 2018. This subset were specific for 2019. And then this subset right here were VOCs that were found in both years. So again, it seems like the types of VOCs that are being emitted in control and treatments are not that different. When we analyze the emission rates, the total average emission rates of VOCs categorized by classes, and in general, sort of like ignoring the seasonal effect and ignoring the year effect, we do see that monoterpenes, sesquiterpenes, and alkane emission rates are significantly higher in some of the treatments compared to the control. So for monoterpenes, we see an increase in emission under heating and nitrogen. Same for sesquiterpenes, and then a very similar for alkenes as well. I wanted to know if there was some specific VOC that could be associated to a specific treatment. So we conducted indicator species analyses to see, again, if there was a VOC that could be associated to a specific treatment. And we found a couple, alpha-pinene and beta-thujone. Their emission rate seems to be higher and consistent under heating and nitrogen, and also beta-thujone, which was also, its emission was consistent under heating and nitrogen compared to control. So they're both monoterpenes, and they both have been reported as having antimicrobial activity. So even though the VOC profiles aren't changing that drastically from controls to treatments, we can say that there are a few VOCs that may be associated to whatever's going on in one of the treatments. So I'm really interested in exploring this a little bit more. Again, they've been reported as having antimicrobial activity, but it's really unclear who is producing these VOCs. So in the past few months, I've been doing some competition work. I isolated a lot of fungi and a lot of bacteria from the swan soils, from the controls and from the treatments. And I do want to point out that I do a lot of culture work, especially looking at fungal traits and how they change in response to different environmental stressors. Didn't get a chance to show any of that cool work here, but happy to talk more about that during the Q&A, or if anyone has any questions, they can you can email me. But anyways, in some of the challenges that I've been conducting, I've been seeing some really interesting patterns. So I've been using this special Petri dishes. They have a division right here. And this division is uneven. So if you look at this division at an angle, you will see that this part of the division is higher and this one is lower. So when you cover the Petri dish, this lower side of the Petri dish allows for gas exchange. So even though the microbes are not sharing any resources in their substrate, they are definitely sharing airspace. And what I've been seeing is that there's a lot of interesting interactions between the fungi, bacteria, fungi to fungi, and bacteria to bacteria. And these interactions change depending on the amount of nitrogen that you put in the media or the temperature that you grow them on. So for example, here you can see that this bacteria inhibits the growth of this fungus. And then once it gains enough space, it jumps the division and starts invading the fungal side. So I'm really interested in exploring these interactions a little bit more. So do soil VOC profiles change under warming and nitrogen fertilization? Yes, but in our data, there's a very strong seasonal effect, and differences between control and treatments are not consistent. So what is the role of VOCs in microbial communities under global climate change? And that is to be determined. So as Krista mentioned at the beginning, I started my lab in NMSU in August. So I want to talk a little bit about the future directions and work at NMSU. So I will continue my VOC work, and I am especially, I want to see if VOCs are controls of microbial biomass at the ecosystem scale under global climate change. So I'm really interested in exploring the connections between microbial communities and VOC emissions. So in the future, I'm hoping to simultaneously measure shifts in the microbial community and volatiles. I will also continue the work related to some of my findings in Alaska. So is global climate change favoring pathogenic fungi in dry land ecosystems of the Southwest? So I want to look into this phenomena of pathogenic fungi increasing under global climate change environmental stressors, but in the Southwest of the US. And I am especially interested in melanized fungi, which there's a lot in desert soils. And finally, I'm going to continue with my fungal trait-based work. I'm especially interested in looking at which fungal traits may be under selection in the wake of global climate change in soil fungal communities in the Southwest. And again, here, I'm really interested in melanin and some other stress-resistant traits. And of course, I want to see how all of these processes and dynamics affect the cycling of carbon. So to summarize, global climate change is affecting fungi from genes to ecosystems. I hope I convince you that fungal responses to environmental change impact biogeochemical cycles, such as disrupting the carbon cycle, and that studying microbial responses to global climate change allows us to understand mechanisms behind ecosystem scale observations. I would like to thank all of my funding sources from both the US and Mexico. I want to thank the people that have done lab work and have gotten funding for the data at the Joint Genome Institute to be available to the public. And I also want to thank my two mentors, Dr. Sarita Fry, Dr. Kathleen Tresier, all of my collaborators in all of my projects, and then all of my undergrads who've done a lot of the work that I presented here. And with that, thank you, and I'd be happy to take any questions.
Thank you so much, Adriana. That was really exciting. So does anyone have any questions?
I do.
I figured, Biddy. Go ahead.
So I want to hear a little bit more about the pathogenic fungi that were that increased under warming. What groups in particular increased, and why do you think they increased?
So there were a lot of transcripts that we could trace back to the Dothidialis. Because we had transcript data, we couldn't, there's no way that we can link the transcript to a very specific taxa. So they were a lot of Dothidialis. Many of those fungi are melanized fungi. Many of them can be saprotrophic, but can also be pathogenic, especially to plants. So I'm not able to tell you exactly which taxa those were because we worked with transcripts, unfortunately.
So do you have any idea of why they were increasing? Could you pick that out of
your... Yeah. I think it probably has to do with the fact that many of them are just resistant to drying conditions. So many of them are just able, to withstand, water stress. I think that was probably one of the main factors why those were transcribing more under warming.
That makes sense.
Okay, and then there's a question in the chat from Monica. Not sure if you can see that. Monica's asking, is there a study on the effect of fire on this fungal community? And then which groups or guilds are probably fire resistant or fire adapted?
There was a graduate student in my lab. She was finishing her PhD when I was starting, and I know that she did a chrono sequence on like a fire chrono sequence on different sites in Alaska. She didn't specifically look at the site that I worked on, but there was a site very, very close by. I don't recall exactly what her findings were, but you can find her publications under Holden or Dooley. If you go into Kathleen Tresier's website, the publications are there. I do know that I have a special love for a fungus, Neurospora, and that fungus, it's really interesting because its spores will germinate only after fires. And one of my PhD chapters were looking into adaptation of Neurospora to warming. And I do remember that Sandra found Neurospora in her fire chrono sequences. I think that's pretty much all I can say about the fire and fungal community. Oh, and then is the petrichor smell fungal VOC? I think that's bacterial. I'm not sure if fungi produce it too.
I had a question.
Yeah.
your Harvard forest study system, do you see shifts in the relative complexity of nitrogen cycling genes in terms of the substrate they target in the high versus low end deposition plots?
I don't know. That's a great question and something that I'd be interested in looking into. Yeah, we don't know.
Peter.
So it was really interesting that you saw such big differences in the VOCs. I was wondering how much correlation there was between like at a plot level, like between plots of the same treatment or plots, same plot.
Yeah, so there was a lot of differences between the plots, even so like the within error was actually varied. So in some cases, we basically found the same thing. So depending on the season or the month that we sampled, sometimes like the within error was significantly different, and sometimes it wasn't. So we didn't really find any consistencies on that.
All right, well, I think it's time for a quick break, and then Adriana will join us at the reception and tea time.
Yeah.
Can I ask a question about alpha-pinene?
Yes.
So alpha-pinene is one of those things that shows up in all kinds of plant compounds.
Yeah.
Like in mints and in nectars and in flower fragrances. And I'm pretty sure, if my memory is right, it's quite anti-insect in a lot of